Hi everyone, you're welcome to my YouTube channel, Everything Medical. On today's video, I'll be treating glycogenesis under metabolism. We have done videos on Pannas pathway and HMP shunt. Endeavor to go and watch it and subscribe and share to your friends. Now, without wasting much of our time, let's do a little background study on a later concept on glycogenesis. As you can see here, this is glyco, which means glycogen. And here is genesis, which means synthesis. Automatically, it's telling you that it is the synthesis of glycogen that is on the latter concept of glycogenesis. Now, let's delve into the class proper. What is glycogenesis? Glycogenesis. Glycogenesis simply means the synthesis or the production of glycogen from glucose. That is the concept of glycogenesis. Then what is glycogen? Glycogen is the storage form of carbohydrates in animal, while carbohydrate is stored in plant as starch. Then this glycogen is very, very essential. It is a fuel reserve for the body that is needed for tissue respiration during starvation state or fasting state. That is how it works. Okay, now our next outline that we'll be looking onto under this glycogenesis is the organ of metabolism. That is the organs in which this pathway takes place. We have the organ of metabolism, that is organs. The organs that are involved. What are the organs in which this pathway takes place? We have the liver and the muscle. Here are the two organs in which glycogens are being stored during welfare state. That is when you have eaten, because excess carbohydrates are stored as glycogen in your body, which is being mobilized during starvation state or fasting. Okay, since glycogens are stored in the liver by a process called glycogenesis, and as well in the muscle, the same thing as glycogenesis, automatically we have liver glycogen and muscle glycogen. So we have liver glycogen, and muscle glycogen. Important to note, the liver glycogen tends to produce or replenish the blood glucose level. That is why the liver is the only organ that influences or replenishes the blood glucose level, because the glucose that is being produced during glycogenolysis to give you energy during starvation state is essential to replenish the blood glucose level. But then, in the case of muscle glycogen, the muscle glycogen is not used for energy production in tissue respiration during starvation state. The muscle glycogen is used as a fuel reserve for ATP production during muscular contraction. Why? Because there is an enzyme that the muscle lacks during glycogenolysis. That enzyme is called glucose-6-phosphatase. The enzyme does not have it. That is why the liver has that enzyme, and that is the main reason why the liver tends to replenish the blood glucose level, because it contains that enzyme during glycogenolysis, that is mobilization of glucose from glycogen. Because the one we are doing today is the production of glycogen by glucose. So during welfare state, that is glycogenolysis, whereby glycogens are being broken down to give glucose, the muscle glycogen is not useful to produce energy, that is glucose. But then the liver glycogen tends to be useful to produce what? Glucose, because it contains an enzyme called glucose phosphatase. That is why we call it hepatic glucose phosphatase enzyme. Okay, that is how it works. Then in respect to the storage form of them, because the glycogen is being stored as liver, we call it liver glycogen, and the glycogen is stored in the muscle as a muscle what? Glycogen. Okay, these are their percentage. The glycogen stored in the muscle is 2 to 3% of glycogen is stored in the muscle. 2 to what? 3% of glycogen is stored in the muscle, while 6 to 8% of glycogen is stored in the liver. Have you seen it? But due to higher muscular mass, that is numerous muscle in the body, the quantity of glycogen in the muscle is bigger than the quantity of glycogen in the liver. Automatically, the quantity of glycogen that is stored in a muscle ranges from 300 to 400 grams. This is because of higher muscular mass, because the liver is just one organ situated in one place in the body, whereas the muscle is scattered all over the body. That is why the mass of glycogen in the muscle is way bigger compared to the mass of glycogen in the liver. So the liver stores 100 grams of muscle. That is the liver glycogen. It stores what? 100 grams of glycogen. That is the liver. This is how it works. However, we have what we call the fuel reserve of the body. The fuel reserve of the body. The body has two fuel reserves. That is the glycogen and the fat. Because on a welfare state, glucose is being what? Stored as glycogen. And as well, glucose is converted to fat and stored as starch in adipose tissue. That is the fuel reserves. So we have the glycogen and we have the fat. This is how it works. Fuel reserve of the body. Fuel reserve of the body. We have the glycogen. Glycogen. That is glycogenesis. Then we have the fat. We have the fat. That is what we call lipogenesis. 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 So these two are the fuel reserves of the body. Glucose has been mobilized from them during starvation state of fasting. Now, recall that during starvation state, the glycogen stored in the liver is being what? Depleted. While during welfare state, the glycogen stored in the liver is being what? Increased. Okay, let's move back to the fuel reserve of the body. We have the glycogen by a process called glycogenesis. We have the fat by a process called lipogenesis. These two fuel reserves works when you are hungry. That is the starvation state or fasting state, so that it will meet up with the energy requirement for tissue respiration. That is how they work. But then, as you can see here, the body prefers the energy from glycogen compared to the energy from fat. Why? Because the energy from glycogen is easily mobilized. It is easily mobilized for tissue respiration, whereas the energy from fat by a process called what? Lipogenesis, that is during their storage. But when they are being mobilized, it is lipolysis. So the energy from fat, it is slowly mobilized, while the energy from glycogen is what? Easily mobilized. That is during a process called glycogenolysis. Another main reason why the body prefers the energy from glycogen is that the energy from glycogen, the full energy from glycogen is very, very good for tissue what? Respiration. And the energy from glycogen can be produced without the presence of oxygen. Whereas the energy from fat can't be produced without oxygen. So imagine when you are in the process of hypoxia, whereby there is no oxygen, it will interfere with the energy production from fat. Whereas for glycogen, it does not require oxygen in order to mobilize glucose. Now, another reason why the body prefers the energy from glycogen is that the brain needs continuous supply of glucose, and that could be from the breakdown of glycogen by a process called glycogenolysis. Whereas for this, which is from fat, it does not supply a significant glucose for brain metabolism. So it supplies a small amount of glucose for brain metabolism. So those are the main reason why the body prefers the energy from glycogen compared to the energy from fat. Okay, next in line is what we call the cellular compartment, because every metabolism have a specific place in the cell whereby it takes place. And we have said it previously that a metabolic pathway or a metabolism occurs in two different places in the cell. One is either is occurring in the cell cytosol or the cytoplasm, or it is occurring in the cell mitochondria. Then glycogenesis is taking place in the cell cytosol or cytoplasm. Automatically, it is what we call a cytosolic pathway. And since the pathway is occurring in the cell cytosol, all the enzymes are called what? Cytosolic enzymes. As you can see here from my diagram, this is the cell cytoplasm, while here is the cell membrane. So here is the intracellular part of the cell, while outside from this place is the extracellular part of the cell. So this is the cell cytoplasm, while here is the cell membrane. So let's dive into the metabolic pathway of glycogenesis. First of all, glucose does not just jump and enter into the cell of a particular tissue without being transported. So, though after glucose has been digested, being absorbed and enter the bloodstream, going to the specific tissue that is going, it requires a transporter for you to enter that particular tissue. So here is what we call the cell cytoplasm of a hepatocyte, because this is the cell cytoplasm of the liver cell called the hepatocyte. So let's dive into how the glucose enters the cell cytoplasm of the liver hepatocyte. Okay, here is glucose. We have glucose. We have glucose. Okay, glucose enters the cell cytoplasm for it to be broken down and phosphorylated. Okay, it goes this way. This is the glucose transporter, which is the GLU2. GLU2 glucose transporter is a transporter of glucose that is specific in transporting glucose to the liver cell. We have numerous glucose transporters from GLUT1 to GLUT7, but there's no GLUT6. So this is a specific glucose transporter called GLU2. So this is GLU2 glucose transporter. GLU2 glucose transporter, which serves to transport glucose from the bloodstream to the intracellular part of the cell cytoplasm of the liver hepatocyte. Okay, it goes this way. It goes like this. Now, glucose has been transported. As you can see here, this is glucose. Once glucose is transported into the cell cytoplasm of the liver hepatocyte, it will be phosphorylated by a specific enzyme. Although there are isoenzymes that do that phosphorylation. One is in muscle, one is in liver. So it goes this way. Glucose will be phosphorylated to form glucose 6-phosphate. There are two enzymes which are isoenzymes by themselves. We have what we call the exokinase for muscle, and we have glucokinase for liver. That is why it's called hepatic what? Glucokinase. We have glucokinase. So it's liver. Why do we write muscle and liver here? Because this pathway takes place in two different organs. One is the muscle, as we have treated, and one is the liver. Okay? Is how it works. Since glucose is phosphorylated, it requires the usage of ATP. That is one molecule of ATP. Recall from it in Panhardt's pathway. That is glycolysis. It goes this way. Have ATP will form ADP plus inorganic phosphate. That is the PI. And this enzyme requires magnesium ion. So there is presence of magnesium ion in them. Magnesium ion. So here, since glucose phosphate has been formed, this is an important metabolite. It's an important what? Metabolite in metabolic biochemistry because it features numerous metabolic pathways. What are the metabolic fate of this intermediate or a metabolite? Number one is glycolysis. It enters glycolysis. It enters glycolysis. It as well enters the HMP shunt or the pentose phosphate pathway. Pentose phosphate pathway. And it enters what we call glycogenesis, which is what we are treating today. So these are the fates of glucose phosphate. These are their fates, glycolysis and HMP shunt. Okay. Now, this glucose phosphate will be converted to glucose 1 phosphate by an enzyme called phosphoglucomutase. Glucose 1 phosphate. Glucose 1 phosphate is an isomer of glucose phosphate because if you watch, glucose have 6 carbon atom. So there is an attachment of phosphate group at the carbon number 6 in glucose to form glucose phosphate. Whereas in this one, there is an attachment of what? Phosphate group at the carbon number 1. That is why it's bearing glucose 1 phosphate. So this intermediate is an isomer of glucose 6 phosphate. So it is catalyzed by an enzyme called phosphoglucomutase that does the catalysis. Phosphoglucomutase. That is a very, very profound enzyme. Now, since we have formed our glucose 1 phosphate, another intermediate will be formed, which is what we call UDP glucose, uridine diphosphate glucose. We generate what we call UDP glucose. This is a very, very important intermediate in glycogenesis. Automatically, we are going to be arriving in our glycogen formation. Here, this pathway or this step is being catalyzed by an enzyme called UDP glucose pyrophosphorylase because there is a presence of a specific energy bond in it, which I will be telling you guys as we proceed. So this is how it goes. UDP will give us our PPI. That is inorganic pyrophosphate. Very, very profound energy bond in terms of metabolic reactions. So this step is being catalyzed by an enzyme called UDP glucose pyrophosphorylase. UDP glucose pyrophosphorylase. release enzyme. That's how it works. It's very, very important energy bond. Okay. Once our UDP glucose has been generated, we are going to generate what we'll call the elongation or the straight chain of glycogen before forming our branching chain, which is being catalyzed by a branching enzyme. Okay. We generate what we'll call alpha 1 4 glycosyl units. Glycosyl unit. This step is the rate limiting step of glycogenesis because it is being catalyzed by an enzyme called dephosphorylated glycogen synthase, because the enzyme is active in a dephosphorylated state. So it goes this way: glycogen synthase. What does it do? This enzyme tends to add a glucose moiety by forming alpha 1 4 glycosyl units. That is how it works. So here is our step 1. This is step 1. This is step 1. This is step 2. This is step 3. And here is step 4. So move over to our step 5. Step 5 is being catalyzed by a branching enzyme, which forms the branch chain of glycogen. So here it works by forming the glycogen itself. So here we have branching enzyme. Branching enzyme. That is the enzyme that does it, which forms the glycogen by forming the branching chain of glycogen. It forms what we call glycogen. Glycogen. Here, glycogen is a polysaccharide. It contains an elongation chain or the straight chain and the branching chain. So we have the first, which is the straight chain or the elongation chain, which is alpha 1 4 glycosyl unit. That is the straight chain. Straight chain. Have the branching chain. Branching chain, which is alpha 1 6 glycosyl unit, because it is a profound carbohydrate, that is a polysaccharide. Okay, now let's delve into the regulation of this important pathway. This pathway works during welfare state. It does not work during starvation. So automatically it is being favored by insulin and inhibited by glucagon and epinephrine. So insulin favors this pathway by activating glycogen synthase to be in a dephosphorylated state, because this enzyme, that is the rate limiting enzyme of this pathway, works during when it is in the phosphorylated state. That is when it does not have any phosphate group. So this is how it works. Regulation. We have glycogen synthase A. Glycogen synthase A and glycogen synthase B. Glycogen synthase B. Okay. As you can see, my arrow Okay, so glycogen synthase A is phosphorylated. So there is addition of phosphate group inside it. Why glycogen synthase B is not phosphorylated? Automatically, the glycogen synthase A, which is phosphorylated, is an inactive glycogen synthase. So it is inactive. It is inactive. It is inactive. Why the one that is dephosphorylated is active. Active. But then there are hormonal regulations of this pathway. Recall that insulin and glucagon are countercurrent hormones. So when you are in a welfare state, that is what we call hypoglycemia. So there is high insulin glucagon ratio, high insulin, low what glucagon ratio. That is what that is happening at that system, because automatically you are in a welfare state. There is storage of glycogen by a process called glycogenesis. Okay, so insulin favors it. So we put our plus sign. Let's do it this way. Insulin favors it. So is plus. Why glucagon inhibit it and epinephrine? So we have glucagon and epinephrine. What do they do? They inhibit it. Insulin favors or activate glycogen synthase by keeping it in the phosphorylated state. So it does not allow it to be in what? A phosphorylated state. So the enzyme that converts glycogen synthase A to what? To glycogen synthase B is called phospho or phosphoprotein phosphatase. Is the enzyme that does it. It converts glycogen synthase A to glycogen synthase B. That enzyme is called phosphoprotein phosphatase. So let's write it here. Phosphatase enzyme. So this enzyme, what does it do? I mean, this insulin, it helps to inhibit this enzyme so that it will not keep glycogen synthase B in what? In phosphorylated state, because already this one is what? Phosphorylated. It inhibits it. It does it. So automatically this insulin is a very, very profound hormone that regulates this enzyme. So it helps this enzyme to keep this glycogen synthase in a dephosphorylated state. That is the work of this insulin. Whereas this glucagon and epinephrine tends to do what? Inhibit this enzyme so that it will not keep this glycogen synthase in a what? A dephosphorylated state. So let's go. There is an enzyme that converts this glycogen synthase B to glycogen synthase A, thereby keeping it in a what? A phosphorylated state. That enzyme is called glycogen synthase kinase or phosphorylase kinase. So we put GSK is the enzyme GSK, glycogen synthase kinase or phosphorylase kinase, whichever one you want to write, it is there, or phosphorylase kinase enzyme. So this enzyme tends to convert glycogen synthase B to what? Glycogen synthase A, which is the one that does not work because it is already what? Phosphorylated. But insulin will come to do what? To inhibit this enzyme so that it will not keep this enzyme in a what? A phosphorylated state. So insulin inhibits this enzyme. It inhibits glycogen synthase kinase or phosphorylase kinase. So let's write insulin here. Insulin, what does it do? It inhibits it. Insulin favors this enzyme. Why it inhibits this enzyme? These two hormones, which is the glucagon and the epinephrine, does what? Inhibit this enzyme. So these are the regulation of this pathway. Okay, we have done justice to glycogenesis. Endeavor to watch this video to the end. If you have any question, type it on the comment section and subscribe to my YouTube channel for more videos on metabolism. Thank you for watching.